This novel methodology allows us to construct three-dimensional assemblies, adapted from desired individual cells, in an aqueous buffer solution containing non-specific hydrophilic polymer, by establishing stable cell-cell contact. In this video, we will demonstrate how we manipulate desired single cells and construct 3D cellular assemblies without an artificial scaffold. Here, we will show the experimental procedure in a medium with dextran as an example.
To begin, maintain NAMRU mouse mammary gland epithelial cells with 5 milliliters of DMEM containing 10% FBS and 1% penicillin streptomycin in a 25 cubic centimeter flask for 2 to 3 days. When ready to proceed, use an aspirator to remove the supplemented DMEM and add 3 to 5 milliliters of PBS that is preheated to 37 degrees Celsius to wash the cells. Using an aspirator, remove all of the PBS from the flask. Add 1.5 milliliters of trypsin that is preheated at 37 degrees Celsius. Incubate in a CO2 incubator at 37 degrees Celsius for at least 1 to 2 minutes. After this, add 3.5 milliliters of DMEM containing 10% FBS and 1% penicillin streptomycin. Pipette up and down to mix.